Yeah, here we go. Hi, everyone.
Hello, welcome back.
Welcome. Today is our 10th workshop. Wow, man, 10 weeks together working here. So
Yeah,
I'm Felipe Matias, and this wonderful lady here.
I'm Margarita Matano.
And we are working together in this project called PhenomeForce. And in this season of workshops for beginners, advanced people, people that want to learn about phenotyping, called Fridays Hands-On. And we are
Mhm.
really happy for because it's 10 weeks together and we are having fun and learning a lot, right, Anna?
Yeah, actually, yes, I learned a lot, many
And
things that
apply.
I didn't know.
Yeah, and so let's start our workshop today. So if you are new here, uh, subscribe in our YouTube channel
Mhm.
because then you can follow our activities, what we are doing when we start a new live. We have many ideas for next year, and we're going to talk a little bit about that at the end of this workshop, and we can plan the next year together, guys. All right, let's start.
Yeah, because actually it's our last workshop for the 2020. We will take a short break for December, but we are already thinking and planning events for next year. So, as usual, let's introduce us better. So we are PhenomeForce. We started in last February with the aim of providing tools and resources for people that works in plant phenomics. We are like we just started, but we have already many followers in both at YouTube and Slack and also Twitter. But we want to involve more people. So if you want to know more about us, you can visit our website or you can follow us at on Twitter. Also, we have a Slack workplace. It's something less familiar, less common, but we really encourage you to sign because it's a way to communicate with other people. So if you want to join us on Slack, you just send an email at no to phenomeforce@gmail.com and we will add you to the workplace. So this is like for people that are not familiar with Slack, this is the typical window. And here on your left, you have the different channel that you can join based on what you are working on and you are interested in. So we strongly recommend that when you join, you introduce yourself in the introduction channel because then people can know what you are doing, what is your work, and they can also contact you, or you can also contact people based on what you are more interested in. There is the Friday Ends On channel, where if you have any suggestion or feedback, please, we want to hear from you. And also there is the channel suggestions for the next season. We didn't have any feedback yet. So no, we had some feedback by through email, but we want to have more because we are planning for the 2021, and so we want really to do a work that interests most of the people.
And also, Anna, if you have a pipeline, a software, or any
Mhm.
technology that is working well for your group, and you want to share with our community, please send a message. We're gonna schedule a workshop for you, and we can
Mhm.
help you put this information out. The main idea here is share and learn together.
Yeah,
So talk with us.
yeah. So that's a good. Thanks, Felipe, for doing that because we want really to make people sharing their information. So we contact people that we know, but there are, I think, many more researchers in plant phenomics that they develop cool phenotyping setup, and we want um, to share their knowledge and research. So there is also the channel events where where we kind of promote the events in plant phenomics. And as usual, so we remind that um, there is like the registration for the 2021 NPPN annual conference is open. So you please submit your abstract by December 4. We encourage you to register and to this conference because it's a way to know and contact people, but also to let your research like public and to reach many people in plant phenomics, as we did at Phenom 2020. Me and Felipe, we didn't know each other before that. So we strongly recommend you to join this event. And if you are in the Slack channel and you want to know more about it, the same slide is there with all the information. So you can go there and look for the deadlines that they are there. And so even if this is the last workshop of this season, we are very proud of this work. We were not expecting so much attendance. But I think we really enjoy being with you and the speakers and everybody. As Felipe say, we learn a lot. So this workshop series was focused on data analysis in plant phenomics. So we actually focus on open source tool for analyzing data in plant phenotyping. Today it's the last one, and I think it's a kind of a bridge between what we have done so far and what we want to do in the future, because the speaker of today is going to kind of show the pipeline from the beginning through the end, like how you start phenotyping the plant till the end, how you analyze the data. And I think this is something that we think that can really interest the audience in the future. So this workshop series was in collaboration with the NPPN, and we thank Jennifer Clark and Christophe for supporting us. But this workshop series was also supported from the Midwest Big Data Hub. We also want to thank Noah Falgren because he helped us a lot with all the technical issue and the binder environments and everything. So thanks, Noah. We hope we will collaborate with him in the future. And then now it's time to introduce our speaker, and this is for you, Felipe.
Cool. So if you register for our workshop series, you receive a mail with information and a drive with many information. One of that information is this link with steps how you install the software that we are going to use today, right? And you can see that there is many downloads. It's pretty used by our community, and we feel really excited when Larry accepted to be here with us today.
Mm-hmm.
Okay. And if you follow that step, you're supposed to see, actually, you're going to see this interface, which is the software that you're going to use to collect information from roots. And the speaker is going to explain well all these steps. And also, you receive a folder with many information. There is pictures there. There's many examples there that are going to go today and learn how they are using the scanners to collect information. Also, we're going to have we have an R script from where we're going to make some graphics and see some results, which is pretty exciting. And with these, I want to introduce Larry York. He's an assistant professor at Nobel Research Institute. And thank you so much for being here today. He's going to talk about root scanning and image analysis using RhizoVision Explorer. It's a software that they are developing there. And we feel super excited. Thank you, Larry, to be here with us. Let me invite him.
We are to the stream, yes.
Yeah, nice. Welcome. Thank you so much for being here today and sharing your knowledge with us.
Thanks, Larry.
Yeah, thanks so much, Felipe and Anna Rita, for organizing this series. I'm glad it's been a big success for you two, and I hope that my contribution will help people learn a little more about how to phenotype roots. And like you said, we'll be going from actual roots to getting images, doing image analysis to get data. And our RhizoVision Explorer software is sort of designed to go directly from the images to data processing. That's something that we've really thought a lot about in our group because we image thousands of roots, and we like to make it as easy as possible for us and others. So the first thing I wanted to do, though, was give a brief, about 20-minute lecture about roots and root phenotyping. So I'm going to share my screen. So the screen from Maine, Larry, should be up.
Okay.
I made it really hard on these two because I have several computers and cameras going, but I think that they're going to manage very well.
Yeah, I guess it's going to be fun. Let's see.
So we leave you to lead the workshop, Larry. And of course, I want to say that if you guys have any question for Larry, please write them on the YouTube, and we will then ask Larry to answer the question.
Yeah, definitely. Feel free to interrupt me and ask a question live because I think it's more relevant as we go through. That's perfectly fine. So I encourage the audience to ask any questions that they have.
Great. Yes. And also, guys, as we always say, we are using internet, many devices, and if something goes wrong, please stay there. We're gonna give our best to come back in time. All right. So join the workshop, and we'll be right here if you need Larry.
Thank you.
Well, enjoy.
Thanks. All right, everybody. So let's get started with this brief lecture. This most of this lecture comes from a teaching tools in plant biology and is available for free from the plant cell. But I have modified it. This was a project originally that Guillaume Lobe invited me to work on with him, so I'd like to give him credit. I'm here in Ardmore, Oklahoma. This is the campus of the Noble Research Institute. We're a nonprofit agricultural research institute. In fact, one of the first official 501c3s of this status. And my lab is right about here in this building. But I'm down in the basement in our root scanning room. Ardmore is here in the central US. So first, let's talk a little bit about the structure of root systems. We'll go into root system architecture, and let's start with a single root. So root anatomy is fairly complex, but the main things I want to point out to you is that roots grow from the root tip. There's meristem tissue here that expands, so as a root grows through the soil, this is the part that's growing and sensing the environment and deciding what the root should do. And then if you look at the cross section of the root, the epidermis, of course, water and nutrients have to travel across the epidermis and into the center of the root, and that's where all the plumbing is. So the xylem and phloem are there. The nutrients and water have to get into the xylem to be taken up to the top part of the plant, and the phloem is bringing photosynthate down from the shoot that's photosynthesizing. And I'll leave it to you. These slides are available in the download that you received. So if you want to read more, you can do that. So the root system develops by single roots growing, and there's two basic divisions within the plant kingdom, and that is between dicots and monocots. So Arabidopsis or carrots, okra, etc., beans are all examples of dicots. And these are typically what a lot of people would refer to as a taprooted species, and that's because they have a strong taproot. And the taproot or primary root is actually any plant that grows from a seed first grows a primary root from that seed. And that primary root is also called a taproot or the radicle. In dicots, this taproot typically becomes thicker over time because of secondary thickening and laterals emerge from it, and those laterals can branch, thus it can become more complex. Now, if we look at things, the monocots, such as grasses and cereals, even palm trees have this type of root system. They also do start with a primary root, a radicle that comes from the seed. And in fact, this one shows it as short, but those primary roots can grow just as deep, if not deeper. Some of our work at Maze shows that the primary root is indeed the deepest root as long as it lives. But the problem is they often can die or become damaged. Over time, these root systems, as opposed to the dicots, produce nodal roots from leaf nodes. So the same node that a leaf emerges from can also produce these nodal roots. And because monoclots lack secondary thickening, these roots become thicker as they emerge and can become sort of the dominant, in terms of mass, the most massive part of the root system. So two basic differences there. And root system formation is an iterative process of branching. So you have primary roots that branch to lateral roots that branch to even more lateral roots. And so root systems are often discussed as being bushy. So to understand why root traits are important, why are we measuring them, you have to consider the soil where roots live. So where are the soil resources? Where are the roots, and what are they doing? First, consider that soil itself is very complex from a physical standpoint. Soil texture is a major determinant of many processes. And basically, soil texture is determined by the distribution of soil particles. So soil is made from ground up rock over the eons. Rock is ground up to small particles like sand, silt, or clay. So sand is on the coarser side, so higher diameter soil particles, while clay is finer diameter. And so most soils are defined based on their mixture of these two. So you can have sandy, silty, clayey, loams that are sort of a nice mixture, and these type of mixtures are some of the best soils for plant productivity. Within the soil, nutrients have charges. They're usually salts in solution, and soil particles themselves can have charges on their surface. And depending on the exact type of mineral and the exact nutrient, you can have processes like diffusion that's caused by the fact that some nutrients, for example, phosphate is a good example, actually bind pretty strongly to soil particles. And because of this, it sort of acts as a buffer, and they can absorb and desorb. And because of this, the depletion zones for these diffusion limited nutrients will be small around roots, whereas some other nutrients like nitrate can travel by mass flow, which means they travel in the water solution as it moves to the roots during the transpiration process. And so the depletion zone for more mobile nutrients will be larger. So remember that immobile nutrients will have small depletion zones, and mobile nutrients like nitrate will have large depletion zones. So why does that matter? Well, some plants have more lateral roots than others, for example. That's something that we can actually measure. So here you see my spaghetti model with a root system with fewer lateral roots and more lateral roots. So remember that for diffusion limited or immobile nutrients, the depletion zones are smaller. And so it's pretty easy to see from this, this sort of simplistic model where the pink area shows the area of depletion, which would relate to how much nutrients were taken up. The plant with more roots was able to take up more nutrients. So perhaps you would think that more lateral roots are good for immobile nutrients. But now if we consider a more mobile nutrient, you can see a plant with fewer roots and more roots. Well, they have each about the same depletion zone because of the mobile nature of the nutrient. And so you can see the plant with more roots actually didn't acquire more, and that's an issue because roots have a substantial carbon cost to the plant. So we have to think about the efficiency of the root system in the sense of the carbon invested to the benefits gained. And of course, there's many other reasons why plants make roots, such as anchorage or anchorage, and also to have some redundancy, because if they were to be eaten by herbivory or die from disease, there may be a reason why plants actually produce more roots than needed from a nutrient standpoint in order to account for these. So how can we describe root system architecture? Well, there's geometrical information. And these are some of the things that we can get from image analysis, and we'll see today, like depth, width. The convex hull is the minimum polygon that's convex that can be drawn around an object. And the area of this can be an indicator of the explorative power of a root system. But at the same time, these type of features can lead to issues of how to distinguish. For example, these two root systems you see have the identical width and depth, but you can see they're actually very different. So just relying on these isn't enough. Some of these properties can be used as proxies for others. For example, there's some evidence that, for example, steeper root angles would be related to the depth of the root system, like you can see here. So root system morphology describes more specific properties like the diameters, lengths, and orientations of individual roots. So I often think of these as sort of global properties of the whole root system versus more local properties. And really, when you consider a root system, you can think of it as a population of roots or root segments. And so different root classes, like we discussed earlier, different types of roots are emerging over time, and they might be thickening over time. So that means that within a root system, it's actually very heterogeneous throughout it, with different diameters and branchiness and things like that. So root topology describes how sort of the branching structure, how things are connected to one another, because we have the seminal roots, the axial roots is usually what I call all first order roots, so things that emerge from the seed or from the shoot. So those are producing lateral roots, and those can branch. So we can discuss things like the number of those, the distance between those, the angles of insertion, etc. So roots grow over time. So you can see here that a young, small root system is pretty, you can imagine it's easier to measure. And that's absolutely correct. And perhaps that's one reason why so many of us like studies on younger root systems. Because as they grow, they branch and branch and branch. And here's an example of maize from Guillaume's work, where you can see after it's more mature, it's so branched that it's difficult to tell apart the different classes of roots in here, like you can with a younger root system. So that's one of the difficulties that we have as root biologists. And there's other ways to measure topology that I won't go into detail here, but so that you're aware, you can measure the altitude, the magnitude, and different indices to show how complex it is. So for dynamic root system properties, you need time series of images, and ideally you'd be able to follow roots individually. So you could say when a specific root emerges and how fast it grows. Some of these, again, can be proxies. So for example, the elongation rate can be linked to the length at the root tip, the distance to where it starts to form branches or root hairs can be related to how fast the root is growing. There's been an attempt to create a standardized root system markup language that would, it's basically an XML format, so like what's used on the internet and other software to describe hierarchical type of data structures. So this RSML basically creates a text file with the XML formatting where all the roots can be specified, and the idea behind it is that this format would allow greater transfer of data from different data pipelines. So a common data structure for roots. So this is an ongoing development. And now how to acquire root system data. So this is a figure that Guillaume came up with that sort of puts into context different phenotyping platforms for roots with how natural they are, naturalness on the x-axis and throughput on the y-axis. So naturalness is a index based on whether it's 3D or not, whether it's constrained by the pot size, which we know that pot size matters. There's a paper you can look up about that. Local, whether it allows growth, and importantly, whether it deprives oxygen and whether it's soil or not. So you can see the highest throughput platforms are things like agar on Petri dishes. And then the lowest throughput would be full excavation of an entire root system in the field using something like an excavator or a backhoe. Extremely slow and difficult. And there's a lot of methods in the middle. One that we'll discuss a little bit today is shovel omics or root crown phenotyping. that I'll show you our method for that. And from most of these methods, you would have to wash out roots from a media, or else if it's something like hydroponics, you may not wash it out, but you would end up scanning the roots, and we'll also be covering root scanning. So root excavations and trenching go back since even like the 20s with the work of Weaver and others. These are still some of the most detailed drawings of roots in the field that we actually have, because very few people want to dig these type of trenches to do this. And of course, sort of the holy grail, so to speak, the holy grail being something that you wish to attain in the end, would be to be able to know the entire root system structure in situ in the field. But it's very difficult to do. One way to get around this in the field is with this root crown excavation. So you dig up just the top portion of a root system. So it has all the information about where the roots are connecting to the seed and the shoot. It can be very fast, but of course you do destroy the plant when you dig it up, and it's only a portion of the root system. I wanted to show a photo of how we wash out roots in the field. This is important whenever you're washing roots, whether from the field or from soil cores, which I'll show here, and from pots. If you let roots soak in water, it actually helps a lot. So that's why we have about 20 bins here with different roots at different stages. You kind of work yourself around in a circle so that you're always actively washing the roots that have been soaking for the longest time. So that's just a little tip. Mini rhizotrons are clear plastic tubes that can be inserted into the soil, and there's different manufacturers for cameras that can be inserted into the tubes to acquire images of roots growing along the surface of the plastic tube, but still in the soil. These have been difficult in the past, that you can get the data, but it was difficult to process the data because it was mostly done manually on a computer by clicking along the roots. But over the past couple years, there have been some machine learning methods come out for the segmentation step, which is what's difficult in order to tell the roots from the background, which is the complex soil. So there has been some advancement here that I'm keeping a close eye on. Rhizoboxes are similar to mini rhizotrons, but instead they're flat boxes with soil inside that you grow at an angle, so that roots will grow along the surface of the boxes. And then you can image them. And similar to many rhizotrons, the image analysis has been difficult in the past, but I think that these newer methods are giving me some hope that this method could become more useful in the future, including the possibility of doing the image segmentation with more advanced methods like machine learning, but then passing those segmented images to software platforms like our RhizoVision Explorer for doing the image analysis on those pre-segmented root systems. One way to get around this was developed called glow roots, where they use transformed plants that express a luciferase enzyme that can produce light. So you can actually see the root system glowing in the background, which helps a lot. But this requires very sensitive cameras to see the dim light that's produced. Hydroponic growth is very useful for root biologists. I've done several studies with hydroponics, but at some point you usually have to go to soil because it is pretty different from soil, but it has the benefit of being able to easily access the roots, don't need to wash them out, etc. Aeroponics is a variation of hydroponics where you actually have something that's a mister that is, rather than the roots sitting in water, they're in air, but this mister is misting them with the nutrient solution. So you can see these little water droplets there. And it's actually been shown that in some ways this is more similar to root growth in soil than hydroponics. These type of filter paper experiments have been popular the past few years. They allow roots to grow on blotter paper that's wicking up nutrient solution. They grow in a 2D pattern, and they can be fairly easily imaged and analyzed. For 3D root structure, gelum gum has been used that in a clear cylinder that's rotated with the roots inside imaged. These images can be put together to create the 3D structure and analyzed. But again, this methods like this are difficult to implement yourself. And Petri dishes have been widely used for model plants like Arabidopsis because it's sterile. In fact, some people joke it's the natural growth environment for Arabidopsis, but I've been told that Arabidopsis is a real plant that grows in the wild as well, so you can find it in natural environments as well. And we think that part of the future of roots will be non-destructive 3D imaging with X-ray and MRI as well. They each have different strengths, and possibly their combination especially could be a really powerful tool. But the segmentation of roots from the soil can be still difficult with these methods, but progress is being made all the time. One really cool thing with X-ray is that you can actually see the soil structure at the same time. So it may be possible in the future to do studies where you're looking at the roots and the soil structure simultaneously. So how to analyze root system images? Well, this has been helped out a lot because there's a lot of tools that have come out over time. You can look at these slides to see what type of tools are available, but it ranges from full automation to semi-automated. And you can look up these different tools on what is now quantitativeplant.org. And we'll be asking for RhizoVision Explorer to be added to that list soon. We're working on the paper describing it right now. And then once you have the data, how do you analyze it? We often have to do things. I mean, you can end up with 50 different properties for multiple root classes. And so if you try to show all of that, for example, in a paper and make sense of it, it can be hard. So we often do things like add up the total length. So people often just report total root length is one of the major root traits that people report. And of course, it's useful. But then you can get into more detail, like the number of lateral roots, the diameters of different classes, etc. So we can show, in this case, from some of my own work, how the different nodal positions had different numbers of roots or different angles. So you can actually try to find ways to show how even within one root system, these different root properties can have variation. And regression, trying to relate root traits to functions, is this area that I think is emerging in plant biology that I call functional phenomics. I wrote a paper about that last year. And the way that I see it is that we're collecting all this phenotypic data. In my work, I'm trying to go from just measuring like a single trait to measuring tens of traits, including like root traits and shoot traits at the same time. And then the functional phenomics part to me is trying to relate, using this trait variation to relate it to its function and make inferences about the benefits of traits based on their relations to things like nutrient uptake or shoot mass. You know, if a plant, like in this case, a plant with greater branching density had more shoot mass. So does that mean that branching density leads to greater uptake and more shoot mass? This is dangerous because there's also plant allometry. We already know that bigger plants, more vigorous plants with more photosynthetates can support larger root systems. So you really run into a chicken or the egg type question. And so I really think a really important frontier is the data analysis that will allow us to uncover the causal nature of the relationships among traits. Because the traits, this type of phenotypic data is multi-dimensional, so dimension reduction techniques like principal component analysis are useful, and we'll look at a example with some data that we'll process today. Yeah, so that concludes that portion with this brief lecture. So Felipe and Anna-Rita.
Larry, it's already a question for you.
Sure.
Do you want to take it?
First of all,
yeah, we can take it now. Yeah.
Oh, I really like your spaghetti example.
Yeah, I did that while I was a postdoc in Nottingham for my job talk, but literally boiled noodles and made little root systems and took pictures.
As in Italian, I also like that.
Oh yeah.
So the question is divided in two, but I will read it like, okay. So he's asking, what have you seen to be more useful in determining routing ability in dicots? Lateral or adventitious routing?
Yeah, so I'll define that.
As a phenotype that is predictive of success during early development.
Okay. So in dicots, adventitious routing refers to roots that form from the shoot. So also you can call it shoot-borne routing. And not all dicots do this, but I do know my PhD advisor was Jonathan Lynch, and they have work showing that the adventitious roots tend to be finer diameter, so they're cheaper to build. And because they emerge above the seed and they grow typically a little more horizontally, so they're really good for topsoil foraging. So if you have soils that are less fertile perhaps or need more phosphorus, then adventitious rooting can be especially beneficial there. But you have to remember that if you're placing carbon into this shallow root structure, that carbon then is not available for the downward growth of the taproot. So you would expect a negative relationship between carbon investment into the adventitious root system versus the depth of the taproot, all else being equal, but that's not always the case. For example, if you do get more phosphorus, then the plant will grow more, then the taproot will end up growing deeper. So it's complicated, a lot to consider, and I would just study both if it's there, measure it.
Okay, thanks, Larry. So there is another question. There are two questions if you want to take them now or you want to start, and then it's up to you.
I could let's do one more now, and then we'll see how much time we have a little later.
So one, it's about RhizoVision Explorer, and I think we can do it later when you are starting to do that.
Yeah.
So I will do this one. What are your thoughts on dynamic root phenotyping with non-destructive sampling?
Yes, I'm really excited about the future of that with things like X-ray and MRI. Thus far, the work we've seen with those typically actually aren't time series based, which is like the huge strength of it. But I think over the next few years, you'll be seeing more of that type of work, and it's definitely a frontier for root biology. But I also like to point out to people that there's all these like super fancy new technologies, but we can get really far using our bread and butter, like destructive harvest and root scanning. There's so much that we can still learn about roots with these, so don't just dismiss them, like they're in the past or anything. Okay. So
okay. Yeah. Cool. So you can start with the.
Yeah, let's see if I can. So what I wanted to show next was some root crown phenotyping. So these are actually some alfalfa roots that we've had lying around for quite a while. And this is the, let me turn this screen so you can see it. This is the RhizoVision crown platform. It cost about $1,200 to build, uses a monochrome camera. If you look at this paper, all the plans are available, models of cameras, etc. And the way it works is that one innovation we have for this root crown phenotyping, these are roots that were excavated from the field and washed off, like I showed earlier. So we have a clip-in replace system using these boards with a clip. This is mostly hardware you can buy at a home improvement store, including a screen door handle. So you attach the roots like so and put it into this platform. You can't see now, but let's give it some juice, see what happens. So inside here, you may not be able to see it in the back is the root crown. And what I'm going to do is show you that this camera inside, there's a small camera here. Let me get my example of it. So this is a machine vision camera. It happens to be a monochrome camera because if you're not measuring color, you should actually use monochrome cameras because they lack the Bayer filter that's in color cameras and actually will get more precise measurements. It has a small lens, just like a DSLR camera has. It's a fixed focal length lens, but you can adjust the focus and the aperture size to let in more or less light. It has no buttons. Just has one connection, which is high-speed USB type. And so the power and the data transfer all happens through here. And what I want to highlight today is that these kind of sit between things like a Raspberry Pi, which is real popular for phenotyping too, and the use of DSLR cameras. Because what's so handy about these is that I found myself over time that with consumer cameras like DSLR cameras, a lot of people are trying to hack them to, for example, to help to automate the imaging. Whereas these cameras are commonly available and they're made for precisely that. They're made for control by a computer, and they're typically used in factories and other settings for machine vision purposes. So let me ask to share the screen from this laptop here. So you two, am I sharing the screen from the imager now? There we are. So this is the RhizoVision imager software. And it's connected to the camera inside here. Those are the roots in here. This is a live view. Put my hand here. And so this software we designed, it's using the SDK from the camera manufacturer, Basler, and it allows you to adjust some basic settings like the exposure time of the camera, which is of course one of the most important things that we can set. If the exposure is too low, you can clearly see the roots are dark. You'll have a lot of noise in the background. So it's important to really hone in the exposure time so that your images have a high contrast and that they're crisp. Oops, I just set it for like a minute. Okay. The thing I want to do is plug in a barcode. Okay, so what I want to do is tell it where to store an image, and I'm lazy, so I'll just use the desktop. We can set a prefix like group 1. That's basically the same as the file name, and take an image. It acquired the image. We go to the well, that's clearly not the same image. There we go. There's root 1. So this is the image that we just acquired. And now, like I said, this is a live view, so I thought we have a minute here, so I wanted to show a little bit about how cameras operate. So this type of camera, like I said, we can change the focus. So let's put it out of focus. And I think that you can probably see now that it's out of focus. And I'm watching the screen to see when those fine roots are the sharpest. So you can see it's disappearing. The fine root is gone. I'm looking sort of at the top of it. And once it's in focus the most, it has a thumb screw that I lock down. And I want to show too real quick about the aperture. So the aperture is part of the lens that lets in light. And I'm going to turn it now. The aperture is fully open, which sounds good in a lot of cases. You can see, of course, it affected the brightness here. So let me turn down the exposure. So I would usually play more to really try to optimize it, but this is pretty good. So I turned it down from like 10,000 microseconds to 600. So much shorter exposure time. And you also see now that you'd have to refocus as well. One thing that's very different, you might be able to tell that at this point it's very sensitive to how I turn. It goes out of focus way more than when the aperture is fully open. It means that the depth of field is very shallow. And so that means that it's hard to get the whole root system in focus at one time. So actually, you can see I'm just turning it just a fraction here to make it shift. And so for me, that's one reason why I actually prefer to, in this system, to use the aperture closed. So you can see I closed it, and so it's black now. You need to turn up the exposure time again. Because that means I can get the root system, because it's a 3D object, so I can get more of it sort of in focus to measure. And so then we're back to where this would be a pretty good image for image analysis.
Larry, there is a question for the setup. Can visualize and measure fine roots?
This particular system is for root crowns, and some of these roots that you can see in the image are fairly fine. It can pick up roots less than 1 millimeter, which is fine by definition. But if you want, like again, this is for root crowns or shovelomics that were excavated from the field. We'll also show a scanner in a minute if you want to measure really fine roots. You can go up to much higher resolutions than what would be possible here. This system is set up right now to be about 12 pixels per millimeter, which is probably equivalent to something like 400 DPI. And in scanners, we scan at 600 to 1200 DPI, so you'll see finer quality.
Thank you.
Yeah, and then the last thing I want to show with this platform is, like I said, something that our lab has focused a lot on is, well, basically the throughput and the simplicity of going from samples to images, and from the images to image analysis, and from image analysis directly to data analysis, which I hope to show you today. One thing with this platform is that we typically use barcodes for almost any sample that we process in our lab. So just an example of a simple barcode. You can buy a barcode printer, thermal printer, and the exact format of the barcode is not as important. But in this software, we have the ability to enable a barcode reader. And once you enable the barcode reader function, if we scan, we can scan in the barcode to the prefix and acquire the image. Actually, it's supposed to work that it'll automatically image with the barcode. I probably set it up wrong, so I have to look into that. But you can see now that the image with the barcode name is there. And so this allows you, then when you do the image analysis, to bring in this file name into your data analysis. And we do things like use an underscore for the separator so that you can split things into factors like what genotype was it, what plant variety, what was it in a block or a treatment like a high nitrogen pot or a low nitrogen pot. And we'll show an example of that during the data analysis portion. Yeah, so that's all I wanted to show for this camera-based. And this type of camera-based idea, of course, could be extended to, you know, things like the blue paper method or other camera-based root phenotyping. And in fact, this software, it supports color cameras, etc. So you could use it for other applications as well. So you two, I want to switch to the root washing part.
Larry, before switching, there is a question on this setup, so I think it's good that you take it now.
Sure.
Can you use more cameras to take images from different directions to minimize the effect of the angle of images?
Yes. We actually have a version of this with 5 cameras for doing that. It's been in use for a couple years in Missouri, and the developer of this software and our RhizoVision Explorer software for his master's, when I was a postdoc in Missouri, he was working on a master's. There's a preprint, and unfortunately, as you move around everywhere, this preprint was never actually published. Sort of an orphan at this point, but there's a preprint called M-PIP, P-I-P, M, the letter M-PIP, about roots that discusses that. But in this context, what we found out is that if you orient the roots into the widest angle, that image was just as powerful as using the data from multiple cameras.
Mm-hmm. Okay. Thank you.
Yeah, so I'm going to turn off this speaker and mute it. So it'll take me a minute before I can hear again, probably. Okay, I'm back. Can you hear me?
Yes.
All right. So I wanted to discuss root washing, whether it's from something like a solo core or a pot in the greenhouse. This is a crucial step of efficiently and accurately collecting the roots in a sample. Amy Lee from our greenhouse here was kind enough to bring me some switchgrass examples of some plants that we maintain over time. And I wanted to show you 2 things. This has one plant that's grown in a sand-based media that we use for most of our pot experiments, and then one that's grown in potting mix, so like a organic-based potting mix typically used in greenhouses, and a sand-based mixture with perlite and vermiculite, where we can control the nutrient content by using specific nutrient solutions. And I wanted to show you a little bit of washing of each. I'm going to keep the shoots intact because we actually want to replant these to maintain these lines. But of course, if you cut off the roots, it's the same thing. You would usually just cut off the roots right below the shoot. This species has a rhizome, so it's a below ground structure that roots and shoots can emerge from. So I'll put that here. To be really careful with washing, it's good to use sieves, so like a mesh. So I have two sieves. This one is the distance between the wires is about 3 millimeters. It's a number 6, and this one's a finer one. It is a 1 millimeter and a number 18. These are nice because they can fit together. So I actually like to use both. So you put, if you have two like this, a coarser one and a finer one, you put the coarser one on top because that will allow the most of the media or soil, if it's soil. Notice I always distinguish if it's soil or potting media because potting mix is not a natural soil. So this coarser one will allow things to wash through. It'll collect most of the roots. It will be staying in the top, and the finer one on the bottom will collect both roots that pass through and also a lot of the media. But so it's just kind of a insurance that you're not losing too many roots, because of course you are going to lose some roots, and you're not going to collect all of the roots. That's the nature of the work. So this area that I'm using is a sink. You can see it has these type of hoses that are used in the food industry. I know because I worked at Pizza Hut for a time, and I used to have to wash the pans with these. So it's the same type of thing that you would use there. One important thing I'll try to show. Give me a moment to lower the camera. Yeah. This one has a soil trap. So that's what this is. The idea of a soil trap is that it collects soil or media in these trays, and then the water will flow over and spill over and go down the pipes. And then the pipes themselves, because of course you don't want to clog up your pipes with soil. So the pipe itself, you can see the blue pipe has an elbow there so that any soil would get trapped in there as well. So you sort of have multiple places to trap soil and try to prevent it from going into your plumbing. So if you're working at some type of institution, they will thank you to protect the plumbing there. So I'm gonna put this plastic bin on the bottom, and then the fine sieve, and then the coarse sieve on top of that. Let's start with the sand-based mixture here. So this type of thing, people always say they, carefully wash out the roots slowly, and of course that's good to do. Another trick is if you're using something like a soil that's more clayey, just like I showed you a minute ago with those bins, that if you have little plastic cups that you can soak your samples in, like if it's a clayey soil, you can even use soap water, like with dish soap inside the water, if you're not going to do any type of nutrient analysis or anything like that on the roots. That will actually make the clay disperse. And if you let it soak for an hour, then you won't be standing here just trying to spray off like solid clay. So letting it soak is really important. So let's keep washing this off. Let me give you a little bit down view of it. Are there any questions about this step?
There are questions, but they are more on the scanner, so I'm reserving them for later.
Okay.
Okay. Or do you want to take them now?
No, we can do it. If you think if I'll be at that step, this will take a lot longer.
Mm-hmm. Okay. So I will make them later then.
Yeah, so I've almost freed out most of these roots. Like I said, I am trying to keep these alive. If you look at what's on this, there's a few roots there, and so we would typically have like a plastic Ziploc available, or running out of power. Let me get the power adapter. Okay. Am I still live here?
Yes.
All right. So you can see that we still have some roots on here, and so you would have some fine tweezers to pick those off and keep them. If there's still some soil on it, like it's common to kind of just put them in a tray with water, and then you can more carefully swish it around and try to get off any of the small bits, especially or organic matter if it's a soil. And then you can see we have this tray again, and you can kind of just go through it, kind of like this, and look for fine roots. If you don't see many, that's good. If you do, you can actually scrape all this off and put it like in a container, and then put it here. I'll just put it back on top like this, and then you can wash it again to try to see if you can catch more roots. So that's fine in this case. But now I want to show you what happens if you use potting mix. So here's some potting mix.
Larry, there is a question about washing. So how long approximately can the roots soak in water before they start deteriorating?
I mean, a very long time. I mean, relative to how long you would be washing roots, you can store. That's a good question though, about how to store roots more broadly. So, in our lab, we would typically try to take soil cores or pots. If you're not doing anatomical work, if you're not going to slice the root and look at its internal anatomy, you can freeze it in situ in the soil for months, and it's fine. You can store it in a cold room or in a refrigerator for maybe 2 weeks to a month. You might start to see a little bit of mold growing there, so you try to avoid that. But they won't actually start to like rot for a while, and you can actually store the roots in ethanol, 70% ethanol as well, for indefinitely, basically. But as far as like soaking them for this, I would typically soak it for about an hour. So like I showed that picture of the bins in the field, I would have like 10 cups. And then in the first cup, it's already been an hour. So when I take it and wash it, I would put in a new sample into that one and work around in a circle, see? And then whenever you're washing a root, it's already been soaking for quite a while. So good question.
There is also another one. What do you do with the fine roots that break off during washing? Do you keep them for imaging or discard?
I would typically keep them. That's a question to ask yourself. There's a trade-off between throughput and accuracy, and I will say that I have seen people that I thought were too careful, like spending 4 hours on one sample. So maybe if you spend say 10 or 15 minutes being pretty reasonable, I think for a lot of different types of samples, you might get 99% of what's there. And then if you keep working for another 4 hours, you might get the last 1%, say. So sort of a trade-off there in the throughput. And so, of course, it somewhat depends on the effect size that you're expecting. If you're expecting a 1% difference between your samples, well, A, that's probably not a big effect in the first place, but B, that means you need to be more careful, right? So there's definitely a trade-off there. But in general, if you know the roots are there, and try to get it for sure. But after it's getting down to like you have this huge amount of roots and there's like one tiny root left, that's probably not going to ruin your data. But yeah, that is something to think about.
Okay, thank you.
Okay, so let's see what happens when we do this potting mix. Do you think it'll be better or worse? Let's see. So from my standpoint, I feel like I've already washed it out quite a bit, and there's still a lot on the inside. And in fact, I can't really pull this apart. The roots are so enmeshed with the potting mix. And this is a case where soaking it, I don't think would really help, because this isn't about like the potting mix so much sticking to itself as you would find with like clay soil. It's more about that the roots are just tangled up in it, and they can actually grow through potting mix. After you're done, you would find that you still have potting mix or roots inside the potting mix, like growing through chunks. Here's a large chunk that had roots growing inside of it. And so basically, I would say for root work, don't use potting mix. Just flat out, I would say there's really, I don't know many good situations of like why you would really have to use potting mix for your root work. If you're doing pot studies, alternatives are this type of sand, perlite, and vermiculite mix. And people have also explored using products like turfus as a media. And these will really help you to have a much easier time to process washed roots. Are there any questions about potting mixes and things like that?
There is one question, I think, but I don't know if it's related with pot. What is the sample size, number of genotypes that is possible can be to handle with this system? And he's continuing to say, has root starts getting degenerated beyond flowering stage?
Okay. Yeah, so their question, I guess, about when you would sample versus when you would process them. So we use strategies where you decouple most of the steps. So like you wouldn't be trying to like go to the field, get a root, go back, wash it out, go scan the root and do the image analysis before you go back to the field for the next root or something. So if you're looking at, we sample all plants at the same time. Sure, you want to try to do that as much as possible. And, with root work, getting a team together is more or less essential. Root work is hard, I'm not gonna lie. I mean, you have to have hands on deck. In our group, we're lucky here that my collaborators at Noble will work with me. Staff from other labs will work with us. But so more hands makes for a more happy time. As far as the numbers, for phenotyping, I think that we should do more of this with phenotyping to express throughput in terms of person hours. So what can one person do per hour? Lack that data for almost any phenotyping platform that I've seen, but we typically do try to put that in our papers, if you would. And something like this, depends on how big the root samples are. If it's something like potted plants, I think that you could harvest 100 or more in a day with one person, and then watching them out and scanning scanning, really, you'll see in a minute, is probably the main bottleneck.
There is another one, I think,
Sure.
good too.
Let's do one more.
Will topsoil be considered potting mix, and will mixing with coarse sand in 3-to-1 be fine?
Well, I think what is sold as topsoil can be a lot of different products. I think it's often compost, which wouldn't quite be the same as potting mix. Most potting mixes are made from peat moss, and stuff like peat moss that's like large fibrous stuff is especially bad. But yeah, I think that mixing with sand is a good strategy if you want to use a product for some reason. Mixing with sand is good.
Okay, thank you.
Okay, so I'm going to mute and turn off the sound from this one and go back to the main, and then I'll do the root scanning part.
Anna, did you work with roots before?
No, I never worked with root before, but it seems very challenging.
I had the opportunity during my doctor to help friends. It's a lot of work, man. Yeah, but it's great.
I think this. I'm really enjoying this today because you actually see the problems from the beginning to the end. It's very like I'm learning a lot.
Yeah. Maybe, yeah. So guys, if you have an idea about other workshops that's starting from the beginning, collect the data, and to have the analysis, let me know, send a message to us, comment here. And Ahita and I, we are keeping an eye on that and taking notes.
Yeah. Well, there are 2 questions on the scan. I think probably I will wait for Larry to start and then ask them.
Sure. Are you ready, Larry?
I think he's muted. Okay.
Yeah. So I think I can see here. Yeah, that's good. Yeah, you can show the scanner view here.
I will demonstrate.
Have a root sample here. I kind of took the approach from like a cooking show. You know, you put the turkey in the oven and then pull it out, and it's already done. So here's some pre-washed root samples. You can see the scanner here. The scanner is a Epson 12000 XL, and it has a transparency unit. It's a large format for an A3 size paper. The transparency unit essentially creates like a backlight. So it's kind of like the RhizoVision Crown platform I showed before, that the backlight behind the root makes a high contrast, sharp image that's good for accurate measurements and simplifies image analysis a lot. I don't want to spend too much on this step because we're getting close, to more than an hour in. But this is a scanning tray. It's made from acrylic. I can send out a link. Maybe we can post that. I don't know. I think that actually you all might have posted a link to these plans for these trays, and it describes the scanner as well. But basically, with these type of trays, they're waterproof. And we know that we like to use about 400 milliliters of water. Doesn't have to be exact or anything, but it's helpful to know. Let's put in the water, take our root sample, throw it on. I'm going to turn on a light. I think will help a little bit. None of that. Is it better with the light or without?
I guess this way is better. Without the, yes.
Yeah. Great. Let me get my spreading tools. So one interesting thing I've just discovered in doing root work is that most people kind of stumble across a solution for spreading roots where you put pipette tips on the end of something to pull them apart. One important thing is that you don't want to scratch these trays, so don't use metal tweezers and things like that. As an alternative, you can't actually find plastic tweezers. So when you're spreading roots, you're basically just trying to spread them apart. And this is another step where you cannot scan something like this. And that's, I've seen people do it, and then they're like, why can't I get a good measurement? You do have to pull the roots apart so that you can actually see in individual roots. So that means that you just keep pulling apart. This is an area where I think if somebody makes a robot to do this, then that would actually be probably more beneficial for root biology than making like a super X-ray system. So here, I think you can see this is a individual. This is from wheat. So this is either a nodal root or a seminal root. And you can see this one root has all these laterals coming off. So we often try to kind of place such a root by itself and tease apart its roots a little more. Because I'm time limited, I think I won't do that to all these. I'll focus on this one sample. So maybe I'll just put these to the side here. But you would ideally do what I'm doing for all those. And basically, after you get it to this level, you're just kind of teasing out these finer roots and trying to spread them out a little more. If roots overlap like this, I don't know how to see it, like a cross shape, that's more okay. If they overlap like on top of each other, then no software can really distinguish them. But if they cross like this, that's not a huge issue. So focus on trying to make them, not be actually lying on top of each other in a clump. And this part can be tedious, slow. It's good to watch. You can even watch a movie or listen to music while you do this. I want to show you now the scanning software. So we're just using the.
Larry, there are some questions on the scanner. Do you want to take them now, or?
Yeah, I can take it now.
Okay, so the first question that was made like before, I have to find it. So that it's can I use a regular scanner?
That's something that we want to explore more. I'm looking into that. I have a feeling that there's probably a way if you use a good background. One problem with normal scanners is having a shadow in the background. So if you can take it in such a way that there's no shadows and there's a clear boundary between the root and the background, then yes, that should be absolutely fine. I think the trick is just to find out how to do that, and it's something that I'm going to explore more about and that I would share in the future.
Yeah, I think with this answer you already, this was also an answer for another question, so I will make another one. Is there a link to the scanner and trays? Thanks for a great webinar.
Thank you. Did you all put that on the GitHub? I think it might be there, or maybe I can send you the link and you can put it there, or the comments of the video or something, or?
What do you mean? Yeah, you can.
We can put here this description of this video after the workshop.
Yeah, so you can send us the.
Yeah, so if you go to, we put all of our data and R scripts on zenodo.org. If you go to Zenodo, ZENODO.org, I hope I spelled it right.
I will write here.
if you go there, you can look for root scanning trays, and I guess these will come up.
Cool.
And the last one I will make is, have you ever used flat scanners to scan 2D rhizotrons?
Like a RhizoBox. Yeah, I have, and there's an example image in the downloads that we sent. The issue that I've noticed, and it may be worse with flatbed scanners than cameras, was that there's any amount of moisture, such as condensation, like it was humid. Maybe I went from the growth chamber with a RhizoBox and came out to this room where it's cooler, moisture condensed on the glass surface, and it looked okay visually. But once I put it in the scanner, like the way that the bright light works in there, it just made it like a total fog. I couldn't hardly see. So there is some issue there. But maybe a camera-based approach might be better for RhizoBoxes. That's something I'm working on as well. And other people have to.
Thank you.
Yeah, can we go to showing the, let's see. Am I showing this the screen, or? Here's the scanner software?
Yes.
Okay. So, yeah, this is the software that comes with the Epson scanner. Typically, we do a preview first. Close the lid. So I'm going to take off. We don't need the webcam from this one now. Have to close the lid. So the previews are fast. Longer scans at high resolution can take a long time. In the background now, you can actually see a white streak. And I did that on purpose on the previous scan to show that actually you have to clean the area where scans start. You have to clean that area where the scan starts, or if there's like, because I think the scanner kind of calibrates itself to that. And if there's like a dirt particle, then it will leave streaks. So if you notice vertical streaks in a scan, you can fix that by cleaning that area. That's an issue that I've seen. So you can see that we have this scan. It looks okay. Of course we pushed a lot of these roots to the side. But you can see this, and this software, because it has a transparency unit, there's particular settings you use for that. 600 DPI is the minimum for scanning roots, I would say, these days, and people typically scan to TIFF. In my experience, if you scan to a high quality JPEG with minimal compression, that's fine, and I've actually done work by myself to compare the length measurement from a TIFF or a JPEG with absolutely no difference. So in general, maybe JPEGs can be dangerous for plant phenotyping, but I feel pretty confident to say with this type of root scanning that if you're worried about space, that a JPEG is not too bad. And I've also found that you can use lossless compression with TIFFs called LZW compression. You can, with this software, it's a little weird how you change like the file name. It's not as ideal for phenotyping as our RhizoVision Crown platform, but you would change the name here. And then I'll just do it at 300, so it'll be a little faster. You can see options for different quality. So it's scanning now. Don't know if you probably won't be able to see the actual light on the scanner. So you might see it as it comes to the end here. It's right here. You can see the light a little bit, I think.
Yes.
So that's the backlight as well. So here's the one that we just took. So Yeah, you would just typically, like I said, you would do all the sampling first, store it, do all the washing. After you're done with washing, and then do all the scanning. So I do it step by step. And after you wash it, you can freeze the roots. We've done that pretty commonly and haven't noticed any issues unless you want to do something like anatomy. So I'll keep the scanning part short so we can demo the software a little bit without going too much over.
Yeah, so there are questions, but I think so. One is there any issue in using? Oh, sorry. Is there any issue in using JRoots?
Yeah.
JRoots.
Yeah. I don't know how the developers want to say its name exactly. I don't personally have a problem with using it. I had used it a little bit in the past. It has a little different philosophy, like a kind of a project based. I don't think it's actually available, as far as I can tell, to download now. So if you already have it, that's good. But I don't know where you can download it from. Of course, you're welcome to try out, like I showed, the quantitative-plant.org. You can try out different software, of course.
Yeah, and the other one is, have you used the WinRhizo root and leaf scanner? If yes, can you please comment on the differences between the WinRhizo root and leaf scanner with the one you are using here?
I've used the WinRhizo system for roots. So WinRhizo is a product for root measurements that dates back to like 1993. They weren't the first, and they weren't the first to use scanners, but they were the first to make a commercial product. Mm-hmm. Their software can do a lot of the same things. WinRhizo does not produce scanners. They resell Epson scanners. I don't know the current model they're selling. I think it might have been the 10,000 XL. So they sell it, and I think they say they calibrate it. It's proprietary closed source. So I don't know exactly how they calibrate it. We've ground truthed ours with copper wires, and get the right measurements. So it's calibrated in that sense. But basically, what they sell is a similar product as this, and the software is also similar to RhizoVision Explorer. I think it cost about $6,000. But don't quote me. You can ask them for a quote.
Okay, so I think we can move on with the software, and then we will take more questions, I think. Yeah.
Okay, so I'm going to share. Oh, I am sharing. Good. Yeah, so if you open it, this is roughly what it will look like. Oddly, I'm somehow stuck on Windows 8. A lot of you are probably using Windows 10, so there might be some minor differences. So when you open it, this is what you see. This is the menu area, the toolbar, analysis options here. This is the image window where you can interact. Down here, you can see all the features that you can measure. And here's an area that we call the ROI, or Regions of Interest Manager. So that's an overview. Let's look at the stuff that we downloaded for this. There's several examples there. Let's start with a scanned root image. So it's scan3.jpg. So loaded in. You can use, one thing I like about this, you can use the mouse wheel to zoom in and out, and pan just by clicking and dragging to look around. So let's zoom in on an area. It's good to have kind of a close view as you explore the options. So first, in the analysis options, you can see that we can choose between broken roots and whole roots mode. Broken roots is for typical scanned roots like this. There might be other cases. In fact, we found that it's hard to kind of come up with a strict classification for root image analysis. But we have plenty of options to change if you need to. Whole root would be more for things like root crown analysis or the blue paper type things from RhizoBoxes, things like that. So let's convert the pixels to physical units. We can choose between putting it in DPI or pixels per millimeter. I know the scan is at 600 DPI, so you can use that directly, and it's accurate. The preview segmented image button will show you what the default segmented image looks like. You can use display output to toggle between the input and the segmented, or use the spacebar. I'm not very happy with this one because I can see there's some thinning, so it's not picking up enough roots. So we can bump it up maybe to 220. Have a look at that. Even to 230. So this is the segmented image input. You can toggle back and forth. You can see there's a lot of debris in the background, small soil particles, which is fine. This level is appropriate, normal. We can filter non-root objects, and that means you can set it in. If it's in physical units, it's millimeters squared. 1 is generally okay. You can see after I clicked on preview, almost all that background stuff went away. There's still an object here. We can go to the feature extraction pane. It has some options like diameter bends. You can make more or less, and I'll show you what that means. Let's start with 1. You click the play button here for run analysis. And you can see in the bottom, it's output the measurements themselves. And if we zoom in, we can see the red is the color-coded skeleton. And it's from this that we're measuring length. And we also are getting the average diameter, the volume, assuming the roots are a cylinder. One thing that I can say, we accurately measure volume, and it's known in the literature that WinRhizo has a flaw, sort of a silly flaw, really, in how they measure the volume. So it's drastically underestimated when you have samples with variation in root diameters, because they assume they use the average diameter times the length, and an average isn't a good assumption. One thing you can see here is that there's all these little short fake, we often just call them fake roots. And those are common to skeletonization routines. We have a root pruning option that essentially will evaluate if a lateral root is longer than the radius of its parent or not. And I think using about 5 is okay for that, because it's saying, is this lateral root greater than the radius of its parent root plus 5 pixels? So that way you can accommodate some bumps on the surface. If you run this again, you'll see we got rid of all these really short roots. And so I prefer to use this root pruning on pretty much all samples, almost by default, but the user has to do this. You can see when I used it, the number of root tips went from 1,300 to 135. So clearly it has a lot of impact on that. So now let's do 2 root classes. We have this thicker axial root and these laterals. So let's see if we can kind of distinguish those. I happen to know that about 0.3 or so should do a good job. That means that objects or roots below 0.3 millimeter in diameter will show up as red. And you can tweak these until you're happy with it. Are there any questions so far?
No, there was one question before about RhizoVision Explorer, so I think you can take it now. Is there a way to run RhizoVision Explorer on Linux? If not, can you recommend a Linux program with similar features?
So yeah, so currently we don't natively support Linux, but it's all cross-platform packages, OpenCV and Qt for the GUI. So it may be possible to do that in the future. We had to get the software out at some point, so it's hard to support everything. We have had a user successfully use Wine on Linux to run it, and also I think a person on Mac, but we don't support that since it's not made for it. But it may be possible to use Wine. As far as other options, I mean, there's like the ImageJ, what is that called, like RhizoJ or something, but it's not very full featured. It's not in development. But this software is worth the price of a cheap laptop.
Yes. There is another question. Thank you for the wholesome workshop. Is there a way to manually set the scale since we use square plates, Arabidopsis, which are like 1 centimeter grids, and DSLR camera to capture?
There's no automated way. I'll show you how I measure the scale. So you can kind of do it within the software. Here's a picture of a clear ruler. Unfortunately, it's in inches, but well, I guess we can do it in DPI. So one thing you can do is that we support regions of interest, this button here, so you can draw stuff. So actually you can draw a region of interest across like 3 inches, and then it tells you the height is 759. So I know it's 759 dots by 3 inches. So it's 253 DPI. And that means I could set 253 here in the convert pixels to physical units box. And then for other image analysis, this would be a constant. So I'd say the important thing is to make sure that if you're using a DSLR camera on these plates, make sure it's like a constant working distance and such, so that the scale will be the same across all your samples. Then you'll make your life very easy. So getting the right image in the first place is probably the most crucial step of phenotyping. Gridded plates could be an issue, of course. If the grids show up as roots, there's no way to get around that. So.
There are other questions. Do you want to take them now or later?
Let's go ahead and do it. I mean, if that's most helpful. I don't know like what cap you want to put on the length of this. I guess it's already an hour and a half, right? I'm fine to go to 2 hours.
We can do half an hour. Like we can stay 50 minutes, half an hour more. I think you can take a couple of them.
Sure.
So, okay, one is for me and Felipe. It's about if the workshop will be available online. Yes, all of them are available on our YouTube channel, so you can really go back and use them. Because this guy is saying that it's an excellent training for students who would be doing root phenotyping with RhizoVision. Yes, so you can go back on our YouTube channel and rewatch it. So for you, Larry, there is one. Can you measure the angle of the roots, contour area, perimeter, etc.?
Yeah, so we give the network area or the projected area of the segmented roots. We give the surface area, assuming roots are a cylinder. I'll show you now in the whole root mode. We give our version of angles, which could be expanded in the future. So these are the alfalfa cotton root rot images. They're from a published study about the effects of cotton root rot in alfalfa. So the roots were taken from a asymptomatic area of the field, which means it did not have disease, and then from an area of the field where it did have the disease. And you can see a big difference. So with the disease, without. So can the software tell it apart? So let's load one in. This is from RhizoVision Crown. We support rotating the image, and then things like angles will be given correctly. This is a whole root mode. The convert pixels is about 12. I forgot exactly, but it's around 12. These are a lot of the defaults. Preview the segmented image. So this is fine. You could play with it more. Do the image analysis. For the feature output, let's do it about 2 millimeters. That should differentiate this taproot in blue from a lot of the lateral roots. Again, you can play with it more, but so we can go on with the workshop. Let's go. Now, when I changed it to whole root mode and I reran analysis, it deleted all previous analysis because it has different features. So we made a decision with our software that as you, if you change modes or add new features, it erases the data that was there previously. You can save data by going to Save As Features, saves as a CSV file. But so you can do it kind of interactively like that. But we support batch mode, which is what I want to show. So I'm happy enough with this. With the angles, we have average angle orientation here. It's in degrees from horizontal. Our way of doing it's different from a lot of people, and you can argue about. I'm not saying it's better or worse. It's just one way to do it, and so it's clear to be clear like what we mean by average root angle. That's because we measure the angle of every pixel in the skeleton, and then report the average. And then we also report the frequency of shallow, medium, and steep angles. So the taproot, for example, here would be steep, and these would be, like this area would be a shallow angle. And so depending on those ratios, you get the frequency of pixels in these different bins. We have a table that gives the exacts and all that. So that's how we give some information about angle, and it seems like it's useful. And of course, most angle measurements in most software are so highly correlated to the maximum width of the image that it's essentially the same thing. So we also provide width, which can be an index of angle. So let's do batch analysis on this folder. By default, it goes to the folder that you're currently in. We can give a new folder called output within. Let's call this CRR features 2. So we're using it. So this data we'll use in the R script. Let's go ahead and save these. I'll show you what it does. Press start. Cross my fingers, hope it doesn't crash. Kidding. We haven't had many crash reports. In fact, this software is very fast and very stable, potentially 8 times faster than some alternatives because and the developer kind of prides himself for optimizing that. So let's go to here. We can actually look at the output folder. Where did I put it? Oh, okay. I actually used a little different area. So now we should have this. So you can see in the output, you can get these images. For the output, we decided to not do the rotation, but the rotation is in effect for the measurements. But we left it up to the user if they want to rotate the output so that the output kind of matches the input. So here's the data file. Now in the software, one thing I like is that when we convert it to pixel units, we actually, when we convert to physical units, we actually give the right measurements here, the units, millimeters squared. It's all based in millimeters. So if it's a length, it's millimeters, area, millimeters squared, volume, millimeters cubed. But one cool thing too is that to make it, we use R, and of course these are fine for Python or other. So where do you change column width? So we make it kind of R ready using dots as the separators. And when things have units, we actually we encoded the units. So hopefully that's helpful to you as you do your work, that you'll remember, like, what unit was this? Oh yeah, it's the lower root area. Oh yeah, that's millimeters squared. So it's kind of baked in. So that's convenient. The metadata file I'm not going to show here. This tells you all the options that you use. So of course that's good for reproducibility. Another cool thing is that actually these files can be loaded as settings. Load settings. Load it. Boom. So you can actually reuse these and share them. And if you publish a paper, you could share the settings file even. So let's go to, I'm going to copy this features file to the area I'll be using for the R. Yeah, because of the length, I'll stop showing the software. I did want to point out that we also use it for, there's an example of a Rhizo box. You can make this work. You can play around and see if you can segment just the roots for fun. It supports color images. It just uses the red channel currently. We plan to do more work with this. There's a cool new software out called Root Painter that I haven't used a lot myself, but I've seen the output from it and think that it's a good option if you need to do more advanced machine learning for the segmentation. But you can also use it for leaves. So if you draw an area around this, everything here should work. The DPI is 300, so you can scan leaves on a scanner, do the analysis. And the area oh boy. Okay, that's better. There we go. Notice just then, actually, the software didn't crash when the image was all black. Try that with other software. They'll probably crash because they'll get locked up in a loop. So the area here, the network area, is the leaf area. So you can use it for other stuff, but I mean, we did make it for roots. Let's go to R. And because of time, you know, we can't go too in depth. I'm going to use the tidyverse package. Oops. Change this to 2, so it's not the same as what came with you. Okay, I guess I have it in the, will this work? Oh yeah, that worked. So yeah, so one postdoc in my group, Kundan Dakal, showed me that you can use the RStudio API to set the working directory to where the source file is from. So that's kind of a neat trick. So the names, you can see again that, you know, we have all these kind of R-ready names with dots in them and stuff. Summary, things that are numeric will show up as numeric. Characters, you know, will show up as characters. Now remember, let's look at this object. The name of these are stored like this, asymptomatic _1. So imagine that this is sort of the status, disease status, and this is like a block. And so I'm using a string split to split by the underscore, and then take the first column and store it as a new column called status, and then the second part of the split as block. So we can do that. Now, if you look at CRR, you'll see that you have the status and block columns. I shouldn't have done that. The block's actually wrong. I'm not going to try to fix that on the fly. It won't affect our other work. So, I often like to do things like pairs, to look at correlations and how things are related. You can go straight to a ANOVA. You can see that the status is significant for total root length. As an example of principal component analysis here, because it's always multivariate data, so I highly encourage you to try multivariate methods. This is a scree plot, so it shows that 50% of the variation is captured by the first dimension. You can look at which traits are having an effect here. Here's the first dimension. You can see ones with higher values have a higher contribution to that. So that's a good way to know where variation is coming from. But now what I wanted to find out is, like, can this do a good job to differentiate the two disease classes? And the answer is no. Principal component analysis looks for the maximum source of variation, but it's not trying to classify or cluster. So let's try cluster analysis. Now, cluster analysis is a multivariate method that attempts to, and don't ask me about like the matrix algebra involved or anything, because I don't know, but just in general, what it's trying to do is just figure out how can you cluster this group without knowing anything else about the data. How can you cluster it into two groups? Because I told it to try in two groups. So let's see if it can do a good job. And it's not perfect, but you can see in this cluster here, all of the members of it are asymptomatic. And there's some mixture here, but at least for these asymptomatic plants, it can tell a difference. And so we can try to do it with three clusters, and then you kind of narrow it down perhaps, but still not perfect, but you still have this cluster that's, and actually in R, you can look at the images. So what's survivor 8 is in this top cluster. So let's look at it. It's kind of slow to print graphics in R, I guess. So that's what the top cluster looks like. Let's look at this asymptomatic one, would be like this asymptomatic 3 here. What do those look like? Oops, I think I need to do plot new. Maybe the device, yeah, I need to do the device off, I guess. But anyways, you can see that these asymptomatic ones that are so different are, like the taproot is very strong. And so even though we're not, what I think is cool about this example is that we don't try to find or identify the taproot or anything. We measured all this other stuff, and that data can mostly classify these root crowns. And in fact, some of the survivor ones may actually, they're from a zone that was called survivor, but they may actually have never had the disease. We don't know for sure. In this paper, I actually used linear discriminant analysis, where it's more of a classification method, and it could classify with like 75% accuracy. So yeah, those are just some basic examples of what you can do from real output that you have. And that concludes what I wanted to show you today. But I'm happy to take any questions.
Yeah, first of all, Larry, thank you so much. It was really, really nice.
Yeah, I think the audience really enjoyed it. There have been so many questions. So
and many people say thank you and congratulations for the amazing work today.
Thank you.
Yeah. Okay, so I will see if I can find more questions. There have been many. So, okay. How does the software handle broken roots versus connected root system on traits such as length?
Yeah, so in the whole roots mode, by default, I guess I should just go back to sharing my screen. By default, only takes the largest object, because originally we had kind of designed it for root crowns. And so you can see that here, if you put it in whole root mode, it has keep largest component, but you can turn that off, and then you can measure even broken roots in the whole root mode. So that's why I was saying that it gets a little confusing. But the main difference is that the whole root mode can do everything that the broken root mode can do, but it measures like angles and stuff, and angles aren't meaningful with like washed out roots, so we try to kind of separate them. But essentially, root length is handled the same way. It's just whether you filter, essentially it's whether you're filtering out small objects or not. If you filter out other objects, of course, they're not counted.
Okay, so there is another question that I didn't understand very well, but maybe you do. I do not have understanding with some trait like number of crossing. Also, long roots, we have to cut the roots. The number of forks are affected?
Yeah, so yeah, they're using Rhizo parlance, but that's okay. So I think that what you're calling a fork would be equivalent to a branch point, more or less. And number of crossings would be also for us equivalent to branch points. Maybe they distinguish whether it's like a 4-way branch or like a 3 or 2-way. And we haven't done that. I've never found that data to be especially useful. Number of tips is a good trait. Branch points, branching frequency, we do by the number of branch points divided by total length. If you separate things by size classes, you can start to try to say like what the lateral root branching density is, which isn't given by default. Cutting roots, yeah, will affect the number of tips and stuff. So yeah, it gets a little problematic when you start to cut, for sure.
Another one. Can you use different sizes of scanning areas, such as different sizes of scanning trays, or is it just the whole area of the scanner?
When you're scanning, you can use the preview mode to select the area that you want to scan. So you can use different size trays, and the thing that we try to do is once you set the preview, it remembers the cropped area. So our advice is I say to scan only what you want to keep and analyze, so you don't have to crop out the scan borders later. So if you scan the whole area, you would see the borders of the scanner. If you do that in the software, I didn't go into depth really. I used it, I didn't talk about it, but we have regions of interest. So if I just select this one leaf, I don't know if this is still the right, so like this one leaf now is analyzed. So you can crop out a scanner like edges within the software. And in batch mode, like I showed, your region of interest here is honored for batch mode. And it supports multiple regions of interest and naming of regions of interest. And then in your output, you will get those named. Let's do this run. So now regions of interest are called root 1 and root 2. So then you can use, again, directly to data analysis. So yeah, I think that answers the questions.
Okay, I will take other two because they keep coming. And well, most of the people, they are saying thank you for this amazing workshop.
Oh, you're welcome.
Greetings from UK also.
Oh.
From all people across the world. Thanks for your lecture. I have a question. Is RhizoVision usable for time-lapse image series?
Certainly in batch mode, you can process time series data, and then you would have the measurements over time. And we've done that. We've measured root length. This example here with auger plates, we actually did this with a camera over time, taking images every 10 minutes, and we had like root length and over time comparing the two split areas in this image, and to get how the root length was affected by a chemical. So you can use it like that. What you cannot use it for would be like to identify lateral root 1 appeared at day 3 and it grew 1 millimeter a day. You can't get to that resolution of data with this platform.
Mm-hmm.
Yeah, this example is in the drive, right?
This play is it's not time series. If people want to contact me for examples of that, that's fine.
Yeah.
Yeah, actually, there is another guide that it says that I have about 150 images of roots with 2 or 3 different phenotypes. It is hard to cluster them. Are you open for follow-up meeting?
You can send me an email. It's fine.
Yeah, so we can share your email with.
That's fine. Yeah, totally.
There is another question. Does the water in the tray change angles of lateral roots, and does that matter?
Right. So when you scan in a tray and you're spreading out the roots, I don't think that the things like the angles is meaningful. So yeah, I would say it does matter, and I wouldn't use the angle data in that case, unless the roots are rigid. If you can, I have this test. It's called the York test for rigidity, and it means if you take a root crown, and people often ask if you excavate the root crown, are the angles correct? Well, guess what? If you turn it upside down, it didn't change shape. So yeah, the angle is. But if you put it in water and spread it around and change it, then yeah, the angles is not meaningful.
I think we are done with the question. There are people from Morocco, Nigeria that they say, great, they send you thank you. Because one of the other question is like, can RhizoVision measure the roots angle? And you already replied to that. So yes, you can. I think this was a very great workshop. Thank you, Larry.
Well, thanks for having me. Yeah, it was fun.
Yeah, thank you so much for our work and also the amazing tools that you guys are developing. I guess for our communities is really important. Some have a group work on that like this way and providing open source platforms and be able to make this kind of presentation workshop. So thank you. Thank you for being here.
Can I say quickly that we are definitely open to help from the community to further development. This software actually, to me, could be the software architecture can support any type of phenotyping based on image analysis. It's modular and plug-in based, and we are totally open to collaboration. So
awesome.
Yeah.
Thank you so much.
Yeah, and so maybe we will share on, I don't know if we put in the website your email address, but maybe we can add that there. And or of course, if you look for Larry York, for sure you will find his webpage at your institution, so people can find easily your email.
And also the Slack channel, right?
Oh yes, true. Slack channel also.
Cool.
Yeah.
So
yeah, if you look for Larry York roots, then I'll pop up.
Nice. Thank you, Larry.
You're welcome.
Bye.
Thanks for your all's work on this. It's great. A great series.
Thank you.
Bye, Larry.
Bye-bye.
So guys, today is our last day in this season. We had 10 workshops, and it's really amazing. Here, all these workshops it is in our YouTube channel. The next presentation. Yeah, so as you can see, we already have 90 there. This workshop's gonna be there today. It takes like around 24 hours to really stay in our platform, but keep an eye on that. We are really happy because people are subscribing in our YouTube channel. We have over 400 people already following us and sending messages. I guess this is really, really nice. It was our first idea to share workshop because we go in conferences, we know the amazing work, but you have no idea how the people do it, right? So that's why we decided to make this Fridays hands-on workshop series in data analysis. And today we saw a different way, which was amazing, showing all process. Maybe for the next series, we're gonna propose this for our speakers, and it's gonna be great.
Yeah, I think so. Among our ideas, there is we want to do more workshop about the phenotyping setups combined with the analysis. There are many cool phenotyping setups, especially if they are like cheap and easy to build. I think that will be great to promote them. So I think Larry gave us a good example of how we can do it. Maybe we have to make the workshop longer. So
Yeah, have more
especially if
time.
there are so many questions. So we
Yes,
are
that's
very
amazing.
happy for that.
Talk about some statistics of this workshop. We're gonna be presenting this at NEAPPN conference, but here sharing with you guys, we have over 700 inscriptions. I guess it's over 800 now, right, Anna?
Yeah, actually, this morning was like over 800 people registered to the workshop, at least to one of the workshop that we did.
Great.
So,
and you can see that we have a lot of students, postdocs, faculties, growers following our activities, breeders, which was amazing. So thank you everybody for being here with us these 10 weeks. And more statistics. People are really interested on R, Python, C++. You can see R lovers, we are getting it. Yeah, we are really happy with this result so far. And this is the idea about beginners, intermediate, and advanced people. So that's amazing because beginners are looking for information, and as Anahita and I, when we start, and we are on this same page, right, and learning all the time,
yeah.
our focus was everybody, right?
Yeah, actually, we are very happy of this because PhenomeForce is actually the objective is to bring together beginners and experienced people. So we are very happy that we kind of achieved that because half of the people were beginners and after are like intermediate and advanced. So we are very happy of that.
Yes.
And
here I want to say thank you to you, Anahita, for the amazing work that you have been doing. You guys don't know, but she's almost 90 months and pregnant, and she's doing this every Friday here in the middle of this pandemic time.
Thank
And
you.
so I'm really glad to have you being part of this project with me, Anna.
Well,
You're amazing.
we have done a great job together. We are a great team. But what I want to say is that we are very happy because as today you guys show, there are many people from across the world that actually watch this video from the east to the west, and we are very happy and we hope to see you back for the series of 2021. And of course, send us suggestion if you, as Felipe say, if you guys have any tools that you want to promote, please send us information abstract and we will plan a workshop, right?
And keep an eye on our activities. We are going to keep doing this, and we hope for your feedback, and we hope that you guys enjoyed as much Anahita and I we enjoyed this time. And see you soon.
Yes. Bye.
Bye, guys. Thank you.